So thank you again for the introduction. I'm really excited to talk with you today about the research that I've done as a grad student, postdoc, and just starting to do now at the University of Washington. As Dylan said, I'm an assistant professor here. I just started last year, so I'm really still just building up my lab, but that lab is going to continue on the research that I've been building over the last decade or so. So what I'm going to talk to you today about is the role of sociality in cetacean ecology, evolution, and conservation, and that the concept of the lens of sociality and what that does to the ecology and evolution of these species is something that I'm very interested in scientifically, to try to better understand the role that it plays. And so before we get, there we go. So before we jump into the meat of my talk, I just want to give a brief introduction to what I mean when I talk about sociality. So sociality can be, Sociality can be defined as the tendency to associate in social groups and form cooperative societies. And so that can be considered as a survival strategy that is developed in response to evolutionary pressures, and it can have profound impacts on the ecology and evolution of social species. Now, many of you in the room today may be at least familiar with the concept of sociality in mammals, but not too long ago, that wouldn't have been the case. So for a long time, science upheld the idea that humans were the only species that were complex and intelligent enough to have evolved social behaviors and cultures. But in the late 20th century, some scientists began to discover rich, complex social lives in non-human animals, first in primates and then other large mammals, and then small mammals and even marine mammals. And it's not surprising that sociality would exist outside of humans, given the ecological and evolutionary advantages of social behavior. So social behavior can improve hunting and foraging. It can provide improved protection from predators, and it can have kin-selected benefits such as caretaking, and it can also increase mating opportunities. Now, culture is one aspect of sociality that we can all identify with as humans. And culture as a concept is essentially a system of inheritance that defines how conspecifics share information and teach behaviors. So within that system of inheritance, social learning allows information to transfer between individuals. This is really similar to the way gene flow and genetic inheritance work, but those have a different set of constraints. So social learning can happen in one of two ways. The first is that it can happen vertically from parent to offspring, and this is, in this case, in marine mammals, that's mostly from mother to offspring, or it can happen horizontally among members of the same social group. Now, cultural practices that have been stable for long periods of time tend to be transferred vertically as traditions. And because of cultural learning, individuals within a social group will share a number of distinct learned behaviors like diet or language. And those behaviors can modify selection pressure, or may isolate a group of animals from the rest of their species. And if those behaviors are stable for multiple generations, they will be written into the genetic code through evolutionary processes such as divergence and adaptation. And then on the other hand, as those genetic traits evolve, they can reinforce the cultural practices that caused them in the first place. So I'm going to give an example of this process in humans that we can all relate to. So at some point in our history, humans started farming dairy cows and producing milk. And that cultural practice led to changes in our genetic code that allow some of us to process lactose. And that tolerance reinforced the cultural practice of eating dairy. And then that cycle has continually reinforced itself so that today we are a culture that is fully obsessed with dairy products. And that interplay between culture and evolution is what we call gene culture coevolution. And it is of growing interest to scientists who want to understand the evolutionary effects of sociality. So it's also an important concept to wildlife conservation, where identifying and conserving these sociogenetic units within a species is really important to preserving that species' resilience to environmental threats. So sociality is going to have a lot of effects on both the ecology and evolution of a species by constraining associations and information transfer among individuals. So one great example of this is diet preferences. We see that in humans, food is a central pillar of almost every human culture, and it's one of the ways that we define our heritage. Now, research has shown that the same is true in many non-humans, where diet and foraging are socially and culturally learned behaviors. The same is true of a species' vocal repertoire, what we often think of as language and dialect. So for example, in the United States, what I call Coke might be a pop to my friend or a soda to my buddy Steve. And of course, in the United States, we have a completely different way of talking than our neighbors to the north. So I tried to find a Canadian-centric map, and I found a bunch of interesting ones. This one took the cake for me because none of these words actually refers to a summer home. And so I was kind of shocked by all of them. But the point is that everybody talks differently, just a little bit differently, depending on where they came from and who they associate with. And in a similar way, vocal repertoires in non-humans can be highly specialized and effectively work to increase isolation and divergence within species. We also know that sociality is an important driver of mating decisions. We readily see this in humans, where our concepts of adornment and mate finding rituals are culturally defined. But scientists have also shown that sexual selection is a socially driven process in many non-human animals. And our movement patterns are often socially and culturally mediated. And we can track large cultural migrations of humans over time, and that the same behavior can be tracked in whales and many other non-human animals. Now, sociality can also have some negative effects. So, for example, it can be an important facilitator of disease transfer among individuals. And at this point in the post-COVID world, I'm sure that we all know that this is one of the major disadvantages of social behavior across species. Moving on, evolutionarily, sociality can lead to increased genetic structure and faster rates of speciation. So in humans, the selective pressure from social behaviors is linked with the evolution of our large brains compared to other species. And then in other species, specific genes have been linked with the development of complex systems of communication or with cultural practices such as the use of tools. And although these are just a few examples, I hope that they highlight the integral and pervasive effects of sociality on all aspects of ecology and evolution in social species. So today I'm going to tell you about some research that I've conducted on 4 top marine predators, each with different approaches to sociality. So the first thing we're going to do is look for evidence of gene culture coevolution in short-fin pilot whales. And then moving on to killer whales, which are a poster child for socially driven evolution. We'll take a closer look at the socially driven aspects of diet in southern resident killer whales. And then we're going to think about one way in which social behavior can be incorporated into species management using bottlenose dolphins as a case study. And lastly, we'll consider the effect of sociality on health and disease transfer in an endangered population of beluga whales. And so for each of these topics, I'm going to talk to you a little bit about what we know and what we've learned. And then at the end, I'll talk a little bit about what the next steps are in this research. So I'm going to gloss over most of the methods today so that we can focus on the results. But I just wanted to briefly say that the research I'm covering primarily uses genetic and genomic methods with some microbiomics and eDNA. And I also integrate behavioral data, including acoustics, photo ID, and visual observations. And in general, this research takes a highly interdisciplinary approach, and that's because in many cases, cetaceans are rare, elusive, remote, and or protected. And all of these characteristics limit our ability to study their ecology and evolution in meaningful ways. And so the answer to that challenge is to integrate and compare multidisciplinary research techniques that complement each other and fill in knowledge gaps. And so to generate those data, there's an immense amount of work that is first needed in the field to find and sample these animals. And that work is undertaken on vessels that vary in size from 19 to over 200 feet. Tissue samples are collected using crossbow biopsy darts. So you see in the top right an illustration of the use of a crossbow. And in the bottom right, you see a close-up of what a modified biopsy dart tip looks like, and that tip has a small sample in it. Samples are generally roughly the size of the first knuckle of your pinky. Then in the bottom left corner, you can see an example of the way acoustic recordings are collected. Those are most often collected using either surface or bottom mounted recording packages. So in this picture, you're seeing a surface mounted recording package with a short fin pilot whale passing by in the foreground. And then the other really important piece of data that we collect is photographs, which are taken whenever possible in the presence of animals and incorporated into long-term observational studies. All right, so let's dive right into our first example, looking at the effects of sociality on the evolutionary trajectory. And to do this, we're going to focus on short fin pilot whales, first in the Hawaiian Islands, and then stepping back and looking more globally. So short fin pilot whales are one of the least studied cetaceans in our oceans. We know exceedingly little about their ecology and evolution, but we do know that they are deep divers, diving up to 800 meters to forage. They live and hunt in groups of 15 to 25 animals, and they use social calls to maintain group cohesion even while they're diving and hunting. Oh, sorry, just lost connection there for a second. And so since we know that pilot whales have incorporated sociality into their group structure, their foraging behavior, and their communication styles, we want to know, does sociality affect the evolutionary trajectory of this species? Now, this is a huge question, so we're going to start simple. We know that social behavior can have a large effect on the way animals associate. Most social species, rather than associating with other individuals at random, will organize themselves to some extent and show some level of preference for who they spend time with. Those associations can be fluid, or they can be stable for long periods of time. Now, in the case of short fin pilot whales, we found a very temporally stable hierarchical social system in the Hawaiian Islands. Small groups of 15 to 20 animals spend nearly 100% of their time together, and 50% of the time, 2 or more of these groups will cluster together, always with the same groups. And those associations were stable for periods of decades or more. Now, by tracking genetic similarity and relatedness, I showed that kinship is an important driver affecting the organization of these groups. So those smallest groups, what we call social units, are made up of immediate family members, siblings, parent-offspring relationships. Larger clusters then represent extended family groups. And I also found that mating occurs largely within these clusters, which means that social structure affects mating decisions within this population. And those clusters, or extended family groups, each had a distinct acoustic repertoire of social calls, and many of them had calls that were unique within the cluster. So what you're looking at here is a plot that shows one bar plot with the composition of calls. Each call is represented by a different color with one bar for each cluster. And here I just have circles. There we go. I've just put circles on the calls that are unique to each individual cluster. And so in addition to having unique calls within each individual cluster, we found that the whole vocal repertoire used by each cluster is significantly different from the rest of them. So this was one really important distinction between these clusters. And then another important thing that I found was the difference between how these animals vocalize when they're alone with their immediate or extended families, so within those clusters, and how they vocalize when they're in larger groups. So this plot now shows encounters with single clusters as dotted lines, and then encounters with multiple clusters are solid lines. And you can see significant differences across 3 different acoustic metrics, which were the frequency or pitch of the call, the frequency range of that call, and then how long that call lasted. So individuals in large gatherings tended to make calls that were shorter, lower, and simpler than the calls they made when they were alone with their cluster or family. And so these 2 bits of data combined, first, the fact that clusters have unique vocal repertoires, and then the fact that they change their calls when they're with larger groups, Together, these results indicate that short-fin pilot whales use vocalizations as a way to both establish group identity and maintain group cohesion. And it also indicates that vocal repertoire is likely socially learned from other members of their extended family. Now, that level of social organization, social learning of group behaviors, and socially driven mate choice has evolutionary implications if maintained for long enough. So another thing that we know is that these clusters have strong preferences for spatial use. They're rarely seen outside of their core habitat range, despite the ability to travel much farther distances. And so what they've done in the main Hawaiian Islands is divide these into two communities, kind of a southern and northern community. And I found significant differentiation of the nuclear genome among those island communities, which indicates a process of isolation and divergence, presumably largely socially driven, within what is currently recognized and managed as a single population. Okay, so we're going to zoom out now to a global scale. And on this scale, we see similar patterns happening. So the species is found throughout the tropical and temperate waters of the world, and it has the ability to travel large distances. But despite that, we found a process of differentiation in the Pacific Ocean, first using morphological data, and then following that up with mitochondrial DNA and vocal repertoires. And that study was later expanded to use nuclear DNA and examine the whole global distribution of the species. And that research indicated that this global species actually comprises two diverging subspecies, which are called the Shiho and Cise subspecies, and are both in the Pacific Ocean, and a third population in the Atlantic Ocean. And I think it's really important and interesting to note here that the strongest divergences, those between the Shiho and Cise subspecies, happened in the Eastern Pacific Ocean, despite the fact that there is significant overlap in the habit of these two species. So if you look at that kind of rusty orange color and the darker blue, you see that there's significant overlap both in the Eastern Pacific and also up north by Japan. So whatever is driving this divergence, it's likely some combination of social behavior and ecological niche partitioning. We know that this is clearly not a simple story of geographic barriers to dispersal. Okay, so we're seeing some interesting patterns in speciation and geographic distribution within the species, and looking a little bit more deeply into the evolutionary history might help shed some light on what's going on. And so we can do that using whole genome sequences from each of those three lineages that we've defined. Now, I know this plot here is a little bit dense to people who aren't specifically in the field of evolutionary biology, but we're going to walk through it piece by piece. So this is a coalescent analysis. It is basically a measure of genetic diversity going back in time. So the most recent time is kind of at the 0 point on the x-axis, and then you go backwards from there as you move right. And so we have a solid line here for each of the three lineages of short-fin pilot whale. And then what the dotted lines represent are these kind of pseudo-hybrid fake whales that I've made, where I've combined a haplotype from each of the lineages. And I will explain that a little bit more in a second. First, what I want to say is that what we know about genetic diversity is that it's tightly correlated with two things. The first of those is effective population size, and the second is divergence within a species. So looking at this plot, we can glean two important pieces of information. The first is if you look specifically at the solid lines, which represent each of those three lineages, you can see that despite having a global distribution and a large abundance today, the effective population size of each of these three lineages over time is quite small for most of their history. Now, those dotted lines that I mentioned represent those pseudo-hybrid individuals. Those can be used to detect evolutionary divergence between the two lineages that are represented by each of the dotted lines. So in this case, an increase in genomic diversity is actually indicating an increase increasing genetic divergence between those two lineages. So we can use this to estimate when those lineages diverge. So that point in time when those lines shoot straight up to infinity, we interpret that to mean that those two lineages are now completely diverged. So we can say definitively that the Shiho and Nysa subspecies diverged over 100,000 years ago in the Eastern Pacific Ocean. And the Atlantic lineage diverged somewhat more recently. So I continued this line of research a little bit more, and I was surprised to find that despite their recent divergence between those three lineages, there is no significant evidence of post-divergence mixing among those lineages. So recent split, they share the same kind of geographic areas, but they do not mix. What I found instead was that the sister species, the long-finned pilot whale, which is found in the North Atlantic and the South Pacific Ocean, the long-finned pilot whale has been mixing into both the Shiho subspecies and the Atlantic population. And at this point, my working hypothesis for that is that the kind of more gregarious and less structured social strategies that you see in long-finned pilot whales lend themselves to increased association and mating outside of their immediate social groups or even species. Okay, so what we've learned so far is that sociality can have a profound effect on the evolutionary trajectory of a species, both at local and global scales. And what we've traditionally considered to be a single species actually comprises three distinct evolutionary lineages globally, while within the Hawaiian Islands, we've been managing two socially segregated units as a single population. And that level of socially driven genetic differentiation begs the question of what else might be different among these groups. Have they developed specific adaptations, such as diet preference, spatial use, or foraging behavior that will affect their resilience to environmental stressors? From a conservation and management standpoint, it's important to recognize and protect socially driven diversity within a species in order to maximize that species' ability to survive changing ecological conditions. All right, so we only touched briefly on the effects of sociality on evolutionary patterns, but we're going to move on because we have a lot more to cover. So let's look at our second example, which is the foraging ecology of southern resident killer whales in Puget Sound. And we're going to look at that through the lens of sociality. All right, so killer whales are famous among top marine predators for their sociality, so I know I don't have to do too much to sway you here. Among socially learned behaviors, the species is infamous for its wildly variable and innovative group hunting strategies worldwide. But today we're going to focus on the salmon-eating population in Puget Sound. So there are three matrilineal pods of southern resident killer whales. Within those pods, matriarchs act as repositories of a vast wealth of cultural knowledge, and pod members learn distinct vocal repertoires, movement and hunting behaviors, preferences for association and mating, and behavioral adaptations to environmental variability within those pods. And the sum total abundance in this population has not topped 100 individuals in the last 5 decades since researchers started studying them, which earns them the distinctive title of critically endangered and has spurred researchers and conservationists to try to understand everything they can about this population in order to save it. And that clearly will mean understanding the importance of socially driven ecological behaviors in the population. So through decades of research, we've identified 3 main threats and list them in order of importance. And at the top of that list is prey availability. And so to understand prey preferences has been the focus of targeted research since the early 2000s. Now, studying the diet of marine mammals is really tricky. They often forage at depth where we can't see them, and we can't exactly cut open the stomachs of a few hundred specimens to get a representative sample of their prey. So traditionally, diet analyses have relied on opportunistic collection of prey bits and the occasionally stranded animal, which are incredibly valuable data and yet give us only a coarse understanding of what these animals are eating and when. Recent advances in genetic techniques are allowing us to sequence the DNA from fecal samples so that we can ask much more high-resolution, focused questions about diet that will hopefully provide some relevant information to management policy. Now, in the case of southern resident killer whales, what we want to know is there a seasonal or socially driven diet variability component within the population? So first, we're going to take a quick look at seasonal differences. And here we also compared the southern resident killer whales, which are shown in purple, to a healthy population in Alaska, which is shown in blue. And we see seasonal shifts in both populations from Chinook to coho to chum throughout the year. We were surprised to find that in the shoulder season, southern resident killer whales will forage on sablefish, while their counterparts in Alaska are targeting flatfishes. And it's also interesting to see that the Alaska population incorporates a much larger portion of chum into its summer diet than the southern resident killer whales, which may help make that population more resilient to fishing pressure. But we can also expect that there will be socially driven differences in foraging behavior among pods. And taking a closer look at 2 of the 3 pods, we see that the J pod, which is in pink, tends to shift more quickly from Chinook to coho and chum in the late summer and fall. L pod, on the other hand, which is in gray, continues to eat Chinook late into the summer and then switches abruptly to sablefish on the outer coast. And this indicates that each pod has unique differences in its prey preferences, foraging locations, and timing of when it shifts from one prey item to another. Now, these differences will have a group-specific effect on resilience and survival. And what that means, in other words, is that whether or not each pod is able to survive the coming decades will depend in part on whether those pods have the cultural knowledge to find backup sources of food when their primary spots are low. And which pods have the right knowledge at the right time is something that is completely up to chance. So managers are hoping to key into these pod-specific differences to tailor management in the most efficient way possible. And one way that we can do that is by using the fact that salmon are genetically differentiated into stocks according to the rivers where they spawned. So if we can target not only the species, but the stocks and rivers that these pods rely upon throughout the year, the hope of managers is that we can balance the economic and cultural value of salmon to humans while conserving food resources for the endangered and vulnerable whale population that depends on the same stocks. And that work is very much in progress, but at this point we have developed a method that can identify multiple source populations from multiple fish in a fecal mixture. And we are testing the use of that method to identify the specific rivers that killer whales target throughout the year. Results are pending, but in the meantime, what we have found so far is evidence that the different pods have different foraging strategies throughout the year, affecting where they're foraging when, and that they rely on different prey sources during the months when Chinook aren't as available. And that will have important implications to their management in terms of where and when management actions are needed to ensure sufficient prey resources are available. Okay, so we're going to turn now to how sociality within a species can affect our management of those populations. And to do this, we're going to focus on bottlenose dolphins in Hawaii. So unlike the other 2 species that we focused on today, bottlenose dolphins are not considered to be highly social. They do not form stable associations, do not live their lives in groups. Instead, they live in what are known as fission-fusion societies, where individuals form temporary bonds within a large and loose social network. And so a valid question that one could ask is whether, when considering a less social species like the bottlenose dolphin, do we need to incorporate sociality in our research and management plans? And so in Hawaii, bottlenose dolphins are divided into 4 stocks surrounding an island or group of islands. And observations and monitoring of these stocks have traditionally been carried out on the leeward side of the islands, which you can see on this map of bottlenose dolphin encounters between 2000 and 2018. And the reason for that is because it is extremely difficult to observe and photograph animals in the inclement sea states that you get on the windward side of these islands. And this traditionally has been assumed to be an unbiased way to monitor this population because these less social animals were thought to basically distribute themselves more or less randomly throughout their island habitats. So the thought is that if we survey a portion of their habitat, we're going to get a random representative subset of the population. And based on these data sets, we estimated trends in abundance, and we found them to be quite alarming, indicating significant declines in the abundance of several of the 4 stocks in the population over the study period, which was from 2000 to 2018. So here I've shown just one of those stocks, the stock around Maui Nui, and this is just one of the examples. If we had stopped here, we might believe that the bottlenose population in Hawaii is rapidly declining. And that may be true, but a little further probing of the data set showed us that there are some additional important factors to consider when studying this population. And the most relevant to this study is individual differences in spatial use. So in this plot here, purple shows the expected size of an individual's home range if it were randomly traveling throughout its habitat. And then the green bars show the observed size of individual home ranges based on where animals were resighted over time. And what you can see here, what I found was that, in fact, many of these dolphins have very small home ranges, remaining within the same 10 to 20 kilometer area for years, despite having access to much larger habitats. And although we didn't do a direct analysis to determine whether this behavior is socially learned, we expect that it is based on the species' propensity to learn behaviors from their conspecifics. Now, remember that estimated abundance plots for Maui Nui from the previous slide, and notice that this stock showed the most pronounced difference between expected and observed spatial use. And that difference in spatial use, when overlaid with the annual differences in the extent and location of sampling, means that different individuals were identified and recorded in different years based on where the survey effort was conducted. So in most years, survey effort was concentrated between Maui and Lanai, as you see in these plots for 2017 and 2018. But for 3 years, from 2004 to 2006, survey effort extended to include the island of Molokai. And if you focus on the blue line in this plot of discovery curves for each stock, you'll see a significant jump in the number of new individuals identified in the years when survey effort extended into new territory. And in the years following, fewer new individuals were identified, and many of those individuals identified around Molokai were never seen again, which causes the model to think that the population is shrinking. So in this example of the stock in Maui Nui, model estimated declines in abundance may actually reflect the contraction of the study area over time. And the results of this study indicated that new observational efforts covering the range of all 4 bottlenose dolphin stocks would be likely to significantly increase the estimate of abundance throughout the islands, and may shift our understanding of whether these stocks are increasing or decreasing. And so this is an important piece of context for the management of bottlenose dolphins in general and in the Hawaiian Islands, and also for the management of social top marine predators generally. Many social populations exhibit learned behaviors that subdivide their niche space within what scientists or managers may consider to be a cohesive habitat space or population unit. And overlooking that structure can bias our results and lead to management decisions that don't reflect the needs of the population. But incorporating social behavior will improve the accuracy of our population estimates and allow us to effectively address the needs of threatened populations. Okay, so we are moving on to our final case today. And so what I want to talk about last is turning to beluga whales. I want to think about how sociality shapes vectors of disease transfer and can affect population health. Now, beluga's social structure is somewhat intermediate between pilot whales and killer whales, which are highly social, and then bottlenose dolphins on the other end that have social relationships that are less stable over time. Recent research shows that beluga whales do not form stable matrilineal social groups, but they are often found in large social groups with dynamic group membership and many unrelated group members. That being said, mother-calf pairs are stable for multiple years, And that relationship may be one of the primary vectors for vertical social learning within these populations. Now, a population of beluga whales lives year-round in Cook Inlet, Alaska, at the southernmost extent of the known range of beluga whales. And that population has been on the decline since the 1990s, despite measures put in place to protect them, and today is considered to be critically endangered. Managers haven't figured out the reason for this continued decline and are looking at threats to population health as one possible aspect preventing their recovery. Oh, sorry, I meant to scroll through that. So specifically, managers want to know if population health and diseases are inhibiting the recovery of the Cook Inlet population of beluga whales. And so to start to tackle that question, I analyzed the microbiome communities living on the skin of animals in the endangered Cook Inlet population and a nearby healthy population. Now, microbiome research is still fairly new among marine mammal scientists, but we've begun to see trends in the diversity and function of microbial communities that relate to various aspects of a species' ecology, including its physiology, diet, geographic location, seasonality, and health. So when I looked at these two populations of beluga whales, I wasn't too surprised to find that there's evidence of drivers affecting microbial variability on multiple levels. And that included differences among the two populations, which you see here, as well as annual variability, which you can see in this bottom plot. And then getting more to the point, I also found significant microbial differences between diseased and healthy individuals in the abundance of potentially pathogenic species. And in the Cook Inlet population, I identified multiple bacteria that were associated with pollutants, such as activated sludge and industrial wastewater. And while these results are descriptive and not causal, they are important to pointing us in the direction of understanding health challenges faced by this population. And so very little is known about the mechanisms of disease transfer in marine populations and the extent to which sociality facilitates that transfer. We do know from studies of terrestrial species that social structure and strength of associations within a population will facilitate and shape the spread of microbiota and diseases. Now, this is just one example of that, showing that social groups of wild baboons have distinct microbial communities, and that even within social groups, the degree of similarity in microbial communities will depend on the strength of association between the two hosts. And there's also a considerable body of theoretical work showing how different patterns of social behavior can affect the extent to which diseases will spread. So within the framework of this 2017 study that was conducted on socially driven modularity within a population, we might start with the hypothesis that beluga whales, with their highly variable subgroup sizes, have a social structure that creates a high amount of modularity. So look at plot D in the bottom right. Now that in turn is going to increase the number and size of localized outbreaks, but will limit those same outbreaks globally. And so another major area of focus at my lab here is to understand how that kinship in social behavior will influence microbiome and disease transmission in the Cook Inlet beluga. So that is an area of research that my PhD student, Ariel Brewer, is going to be taking over starting this summer. And it's going to be a fun challenge for her and for our lab because it's going to involve developing and improving our methods for microbiome research in top marine predators, as well as deepening our understanding of the role that social behavior plays in the spread of diseases and other factors of population health. So stay tuned for her work over the next couple of years to see the results of this study. Okay, so we've seen 4 examples of the way sociality can interact with the ecology, evolution, and conservation of top marine predators. So just to wrap up really briefly, I want to leave you with a few parting thoughts about the importance of this kind of research. Now, the first thing I want you to remember from our time today is that social behavior is an integral part of the evolutionary ecology of top marine predators. It can increase rates of divergence in speciation, and it can facilitate speciation in sympatry. It can also drive specialized behaviors such as diet preference that increase a species resiliency, but can also make an individual or group more vulnerable to anthropogenic threats. It can affect ecological behaviors in ways that will bias our understanding of the population if not accounted for. And finally, it can affect the rate and pattern of disease spread throughout a population. And these are just 4 of many, many ways that sociality affects the evolutionary ecology of a species. And so the flip side of that picture, and the second thing that I want you to remember today, is that understanding social behavior is important to the conservation of these top-known predators. I would argue that you can't actually conserve these species without understanding their underlying social processes. That will allow us to maximize the genomic and cultural diversity in order to maximize species resilience to environmental stressors. It'll allow us to use known social behaviors to inform population models to ensure accurate management plans. And it can help us to understand the ways in which social behaviors can increase risks faced by individual populations. And so that wraps up my talk. So before I open up to questions, I just want to take a minute to acknowledge that this research has been made possible by so many collaborators from many, many different organizations, both within the United States and internationally. And I'm grateful for all of them to helping make it happen. I also want to thank various funding organizations that have supported my research to date, again, both nationally and internationally. So thank you all so much for your time and attention today, and I am happy to answer any questions that you have.
The question I wanted to ask was back when you were showing us that graph with all the different whale calls, and you put a circle on all the unique ones. And I noticed that the group H20 didn't have a unique call. So what does that mean in terms of their sociality? Like, is that significant?
That's a great question. We don't have enough data at the moment to know whether or not that's significant. It could mean that it's a new group that recently broke off from a different cluster, and so it still shares some of its enough of its calls with other clusters. Or it could just mean that we don't have enough data on that group.
Awesome, thanks.
Yes, thank you. Thank you for this great talk. Just a quick question. I was wondering, how do human activities affect the social or sociality of species? Is there any evidence of that? Could be either fishing or transportation or spatial limitation, etc. Yeah, thank you.
Yeah, okay, so just to make sure I understood the question, I think you asked whether human activities affect social behaviors in species. And I
Yes.
don't think, okay, I don't think that I've seen specific evidence that human behaviors are shaping social behaviors. That doesn't mean that it doesn't exist, just that we don't have evidence of it yet. What I have seen is there are a couple different publications that show that social cetacean species are likely to be less resilient to anthropogenic stressors than non-social species. And that's kind of because that that effect of, you know, this group does one thing and this group does another thing. And so this group, you know, if there is some kind of a stressor that this group is vulnerable to, it's likely to wipe out the whole group and not just one individual.
Sure. Thank you
for your talk. I was just wondering if whales or other marine mammals form mixed species groups, and if so, what do you expect? How will you expect that will affect your predictions of parasite transmission or evolution?
You do get mixed species groups occasionally, quite rarely. So there are a few examples of mixed, I wouldn't call them mixed groups necessarily, but like, you know, one animal from another species that joins a group from, you know, so one narwhal joining some belugas or one beluga joining some narwhals. And again, this happens very, very rarely, so I don't expect that it's going to have a huge effect on kind of a species-wide basis. But in that case, my, you know, what I would imagine happens is that the microbiome of that one individual becomes more similar to the microbiome of the species that it's traveling with than to its own species. But that's a great question. There's evidence that suggests that there are species-specific core microbiomes that are affected by that are kind of can be described by phylogeny, right? So the more closely related two species are, the more similar their microbiome. And so there's a lot of research still that needs to be done on to what extent an animal's microbiome is affected by its by genetic inertia, by its phylogeny, versus how much that microbiome is affected by who it spends time with and what habitat it's spending time in. So still an open area of research for sure.
So I was wondering if there's any observed differences in plasticity for traits that are learned versus inherited. I was thinking especially like in the context of management, if a species has some like maladaptive trait, would we be more hopeful if it was like a learned or inherited trait? And then also acknowledging that it's probably more of like a spectrum than a binary for learning and inheritance.
Yeah, well, it's a great question. So it's learned in both directions, right? Inherited behaviors are still learned. They're just learned from parents rather than conspecifics. But I don't know that there's any research on that question out there currently. I think it might be a little bit difficult to address it because it's hard to look at social behaviors in these animals. We can't put them in tanks. We can't manipulate their environment and see how they respond. But it's a really, really great question, and one that I would hope that as we continue to look more deeply into these social behaviors, I would hope that we start to address questions like that.
Hi, fantastic talk. And my question is not fully formed, so bear with me. It's forming. So given the disadvantage of inbreeding, many species will have, social structure where it's only the females that stay together, the mother, like it's a matrilineal, and then the males will either disperse or at least mate outside of the group. But then you mentioned for the short feet pilot whales that they mate within. And so I guess I'm both curious about what's the impact of that? And also, when is there kind of a threshold where like the advantage of sociality like goes over the disadvantage of inbreeding? Is there anybody that studied when that threshold occurs?
This is a great question, and I don't have an answer for you. So in the short fin pilot whales, we found evidence that a lot of mating occurred within those kind of the clusters, right? Those extended family groups. And basically what that leads to is, a high level of genetic similarity, a high level of relatedness within those groups. There is clearly some mating that's happening outside of those clusters. And I did a lot of field work in the Hawaiian Islands over the years that I was working on this study, and occasionally you would find these enormous, just like 75 to 100 pilot whales all in one place. And clearly they're there for social reasons and possibly for mating, but it was rare. And so I think that the very, very first part of that question is the result of that is that genetic similarity and signatures of inbreeding are relatively high in that population. Although they didn't, when we looked specifically at inbreeding, we didn't find significant signatures of them being considered inbred. They just were very highly genetically similar. So the second part of that question is kind of like, what is that threshold? And that is a really, really great question that's really, really hard to get at. There are a couple new studies that have come out recently on runs of homozygosity, inbreeding, and kind of overall genomic diversity and resilience. And one of them on the vaquita, and one of them that's actually, I think, not out quite yet, but coming out in the next couple weeks or a month or so, is on the southern resident killer whales here in Puget Sound. Though they show, they're fascinating because they're very they did a similar kind of research on two very endangered populations and found wildly different results in terms of how inbreeding has affected the resilience of those populations. And so a lot of this gets into how long has this population been stable at the current size, and has it been able to effectively integrate any of those genetic, those potentially deleterious alleles in a way that is safe for that population. The short answer being, we don't know yet. More research is needed. And I think killer whales, especially killer whales in the Pacific Northwest, are a really great place to start, because we do know so much about the survival of individuals within those populations. And so we can start to look specifically at to what degree those individuals were inbred and compare that to their survival.
Thank you.
Mm-hmm.